Hello, my name is Jody Rosenblatt from the Huntsman Cancer Institute at the University of Utah. And in this series of lectures, I'll be telling you about how epithelia maintain constant cell numbers through epithelial cell death and division. And in this segment, I'll be talking specifically about how epithelia regulate cell division. But before I do, I just want to introduce what epithelia are and what they do. You can see that these epithelial cells work together here collectively to form a tight barrier. And this is true not only for your body, which is coated by multiple layers of this skin, but all the organs in your body are coated by a similar skin, but it's only comprised of one or two cell layers. And even though it's only one cell layer sometimes, the purpose is still to perform a tight barrier for all these organs. And because of this, this acts as the first line of defense for any types of toxins or pathogens that may try to enter the body. It's also essential for the form and function of all organs. Without this epithelial coating, you really wouldn't have any proper organ function. So this picture that I'm showing you looks very static, and it looks like bricks in a wall potentially. But what you should really know is that epithelial cells turn over at some of the fastest rates in the body. Those lining your gut, for instance, can all turn over within only 3 to 4 days. So this is lots and lots of cells dividing and dying constantly. And this just shows you one cell that's dying and another cell that's dividing over here. And when you think about this in your epithelium barrier, you can imagine all this cell death and cell division could disrupt that whole barrier function, which is the total purpose of the epithelium. But in fact, it doesn't, and we'll learn how that happens. And moreover, one thing that's very important is to make sure that the numbers of cells that are dying actually match those that are dividing, so that no gaps ever form in that barrier. And the other part is to make sure that the cell division rate does not outpace the cell death rate. If this happens, tumors could form. And nowhere is this more important than in the simple epithelia like these, because this is where 90% of all cancers arise, is in these types of simple epithelia. So what we'll be covering in these lectures is how cells control epithelial cell death and division. In the first lecture, I'll tell you how epithelial cells divide. And then next, I'll tell you how they control cell death through a process that we identified called apoptotic cell extrusion. And then finally, we'll examine some diseases that can result when this epithelial extrusion becomes misregulated. Before I do, I just want to introduce you to some model systems that people have used to study epithelia. So most studies have really occurred in cell culture, epithelial sheets in cell culture. And another popular organism has been Drosophila. And these have been fantastic for a lot of the genetics that have helped us understand key regulatory proteins. Some other growing model systems are zebrafish, which is what our lab uses, and Xenopus. And both of these skins in the developing embryos act as really great simple epithelia that are very similar to the types that coat our organs. So now we'll turn to this problem of how epithelial cells control division, and moreover, how do they link that cell division rate to the death rate? So when we started thinking about this, most of our understanding of how cells divide really comes from a different type of cell, which is the fibroblasts that you can see here, these two fibroblasts here. And the reason for that is really mostly historical. When people took out explants from culture, so they would take out an embryonic explant, these are the types of cells that would start to crawl out. And because of this, they were really easier to culture. So really, because they've been easy to culture, most of the studies on cell division have taken place with fibroblasts. And some of the earliest studies happened with Abercrombie, who was watching these cells migrate. And what he found is that when they migrated, they sort of spread out as far as possible. When they came into contact with one another, they'd stop their migration so they wouldn't crawl over one another. And this he termed contact inhibition of cell migration. Now, this idea of contact inhibition of cell migration also led people to think that maybe cell division was controlled in a similar way, so that cells would stop dividing when they came in contact with each other, but the free contacts were what allowed cells to go on and divide. And this was really led to a lot of the thinking about how cancer cells are not controlled because cancer cells grow on top of each other, and that is called loss of contact inhibition. So the studies on this were actually pretty sparse, and these came really about 20 years later in 1968 in this paper by Holly and Kiernan. And what you can see here is that they grew fibroblasts in culture. These were different types. These were immortalized fibroblasts. And you can see that grown in 10% serum, they'll grow until they get to a point where they level off. And that point they identified as the point where they reach confluence, or they come in contact with one another. And so this really led people to think that this was a point where cells would stop dividing. However, in this paper, Holly and Kiernan also wondered, well, maybe when they reached contact with each other, they had less access to growth factors. And so to examine that, what they did is they added more growth factors. And the way they did this was just add very high levels of serum to these cultures. And when they did so, this is 20% and 30% serum, they found that they could actually overgrow and grow beyond contact inhibition. And so that really, if they added more and more growth factors, they could bypass this. So these two factors have really been critical in our thinking about how cells may control cell division. That when they come in contact with each other would normally stop dividing, but also their access to growth factors has been very important. And this is true in the cancer world too, when we think about growth factor regulation of cell division in cancer. So there's one caveat to all these studies, and that is when we look in the body, what we find is that fibroblasts actually rarely divide. And how do we know this? Well, from a growing trend called tattooing, which I'm sure many of you know. And when you think about tattoos, you think about where does the ink reside? Because if it resided in a tissue like epithelia that are constantly turning over, then these tattoos would fade over time and smear out as they would migrate. But the fact that they don't suggests that they're in a cell type that isn't dividing very often. And what cell type that is fibroblasts. So if you take a biopsy of any tattoo, you'll see, like here, that the ink in those tattoos really comes from, is residing in the fibroblasts there. So these cells are really not dividing very frequently. And it's not that they're static and can never go into division. It's that they could be activated to divide, but the reason that they don't is that they're just static. Now, they can be activated to divide, and that comes from another growing trend, which is tattoo removal. So when you use lasers to ablate these tattoos, what you're actually doing is targeting these fibroblasts for death. And this also causes a wound healing response, which causes high serum levels. And this will replace those fibroblasts by cell division. So this will only happen a few times. And so through an iterative process of laser ablating and causing wounding, you'll eventually remove these tattoos with the dye. So now, when we think about these kinds of cells, we have to think about this may be very different in epithelial cells. Epithelial cells, unlike fibroblasts, turn over at a really high rate. And so what drives then epithelial cell division? Well, we did the similar kind of studies, but we did them with epithelial cells in culture. And here we use MDCK cells in culture, which are dog kidney epithelial cells. And we did a similar type of study to Holly and Kiernan, where we plated the cells and looked at the rate of mitosis every day by phosphohistone staining. And so what you see here is that the cells really never stop dividing. They just slow their division rate. But the point that they slow their division rate isn't necessarily due to when they form contacts, because what we found is they actually become confluent on the second day. But they really start to slow this growth rate much later. So it doesn't seem that the contacts then are necessarily just controlling that cell division. It really seems more that it's the density. And the way we identified that is if we look at the density, you can see that they start to, the point at 5 days where they start to slow this division rate is when they reach this maximal density that they prefer. So this suggested to us that maybe the density could be controlling the epithelial cell division. But if you think about the studies from Holly and Kiernan, potentially at this point where they're very high density, maybe they don't have as much access to growth factors. So we examined this by making the growth factors not limiting. And the way we did that was to exchange the medium every day, so they had fresh medium with fresh sera. And when you do that, you see no change in this rate at all. They seem to slow at the same time. So this suggests then that the real control on epithelia is really the density at which they're grown. And when we started to look in different tissues, we found the same kind of evidence. Here you can see just an example of human colon here, and this is in the crypt where the cells are dividing. What we noticed in this crypt, as well as lots of other epithelia, is that the places where the cells were dividing were always more sparse than the places where they weren't dividing. And in fact, in all the different types of epithelia, we found that they were always 1.6-fold more stretched or more sparse than the places where they weren't dividing. Now, this could just be a result of these cells are bigger and they're ready to divide, and these are just what dividing cells look like. Or it could be that they're sparser and they're sensing this tension. And so to test this idea, we and others looked to see whether we could bring cells that were in this sort of more static state into division state by stretching them. And so this is work done by Stefano Piccolo's lab. And what he's done is taken epithelial cells and grown them to a high density, so they're growing very slowly. And then he'll stretch them. And what he found is that after about 6 hours, they enter the cell cycle. And you can see that they're going through cell division. Well, right now they're starting to go into S phase because they're incorporating BRDU, which shows that they're starting to replicate their DNA. And this is after stretching. So from previous work that they had done, they had a good candidate for what may be controlling this whole process of stretch-induced proliferation. And that came from YAP and TAZ. And YAP and TAZ are transcription factors that work downstream in the hippo pathway that are responsible for proliferation in this pathway. And what you see is that if you knock down YAP and TAZ, that really blocks that proliferation. You can see in this graph here. Now, the reason they thought these might be candidates for controlling epithelial cells, depending on what size they were and how stretched they were, is that they could see that this transcription factor could only be active when it went into the nucleus. And they found that it was only in the nucleus when these cells were in a stretched out state, as you can see here. And the way that they examined this was to plate them using micropatterning prints of different matrix to restrict the space that they could spread out on. And so when they restricted them very much, then this transcription factor could no longer enter the nucleus and it stays in the cytoplasm. So this prevents it from going into proliferation. And when they looked at this in an epithelial monolayer that was grown densely, they saw the same kind of effect, where this YAP and TAZ were in the cytoplasm here, but when they stretched it, it quickly went into the nucleus, and then presumably to activate cells to divide. So another candidate that came recently from James Nelson's lab is beta-catenin. This is another transcription factor that works downstream of the Wnt pathway and is also responsible for cell proliferation. Now, an interesting fact of beta-catenin is that in epithelial cells, they reside at these cell-cell junctions here, bound into this E-cadherin complex. But when the Benham Pyle et al. Stretched these cells, they found that the beta-catenin could rapidly go into the nucleus to activate cells to divide. And when they looked at these two factors and the entry of YAP and beta-catenin into the nucleus, they found they came in different waves. So YAP would, after stretch, would first come into the nucleus at around 2 to 4 hours. And then around 6 hours, beta-catenin would then go into the nucleus. And by using inhibitors of their transcription downstream of YAP and beta-catenin, they then found that these transcription factors played different roles in different parts of the cell cycle to activate different stages of the cell cycle. What they found is that when you activate cells to stretch, then YAP will go into the nucleus and activate cells that were in G0 to enter the cell cycle at G1. And also, later, beta-catenin would then go into the nucleus following stretch to activate cells to undergo this program of S phase to passage through the cell cycle. Now, from doing this, by stretch and YAP and beta-catenin driving transcription, this could activate these cells to undergo the cell cycle and divide after about 12 to 24 hours. When we looked at this process, we wondered whether the cells could respond more rapidly. And what we found is that stretch can rapidly activate mitosis, in fact, very fast, much faster than 6 to 24 hours. And the reason we wondered this was really from movies that we had made. So this is an MDCK monolayer that's been grown to confluence. It's at a high density, so it's turning over very slowly. And when we make a wound, what you'll see is that the cells move in to cover that space very rapidly. And that is the prerogative of an epithelium, is to cover that space so that it provides that protective barrier. Now, when the cells meet, they stop migrating, which is similar to what Aberrant found with contact inhibition of cell migration. But what is interesting is that after these cells close that wound and they're in a very stretched state, they very rapidly divide. And this can be seen more clearly graphically. So now we're just looking at the mitosis rates, or the division rates per frame. And what you see is there's very few cells dividing before the wound closes. But after the wound closes, there's a lag of about an hour or two. And then there's this rapid burst of proliferation that responds. And so we wondered whether these cells were activated to divide just by stretch, or whether they needed to be wounded. And so to do this, we took these cells that were at these high densities and stretched them and tested whether they could enter division or not. And when we did, we found they were able to induce mitosis very rapidly. Within only 1 hour, these cells had really increased their rate of mitosis. And although this isn't a very high rate of mitosis, it's actually only about 5% of the total cells that are undergoing mitosis at the highest rate. It's actually enough to make these cells go back to densities that they prefer. So now if we're looking at the density by the length of the cells, which is a more accurate look at this density, you can see that by 4 hours, when these cells have reduced their division rate, they've gone back to densities that they really prefer to remain at. So how do they do this? What mediates this stretch-induced mitosis that is so rapid? We didn't think it was likely to go through this whole entry into the cell cycle. And so we started looking at candidates in the stretch and stress pathways that may activate mitosis. And what we found was a newly identified stretch-activated channel called piezo-1. And when we knock that piezo-1 down, we see that the stretch-induced mitosis by phosphohistostaining goes dramatically down. So it's not induced to divide following stretch. This was true also in the wound healing results. So this is the knockdown case, and this is the wild type case. And you can see that after the wound closes in the wild type case, we get the same induction of proliferation. But in the case of the knockdown, these cells don't divide after they close the wound. Another interesting thing we found as a side while we were watching this wound closure is that piezo knockdown disrupts the inhibition of cell migration. So you can tell now, if you're watching these two movies together, the wild type monolayers will crawl and close, and once they close, they stop migrating. However, the piezo knockdown cells had no problem closing that gap, but once they did, they kept migrating. And this can be seen better when we track just a few cells in the monolayer. The wild type cells stop at the same time that the wound closes, but the cell migration carries on well after the wound closes in the piezo knockdown. So this was confusing, but it was sort of interesting in going back to these ideas of contact inhibition of migration and contact inhibition of cell division, that maybe piezo could be controlling these two activities. These were studies that we'd had only at this point seen in cell culture, and so we wanted to investigate whether it was true in vivo. And so to do that, we looked at a good simple model for epithelium, which is the zebrafish epidermis. And when we knocked this piezo down using a morpholino, we found the same thing to be true. So now these epithelial cells greatly reduce the proliferation rate. So what we found then is that mechanical strain can activate not only these early stages for cells to enter the cell cycle, but also much more rapidly, where they can activate cells to undergo mitosis. And because this whole process typically takes around 6 to 12 hours, 6 to 24 hours really, we thought it was unlikely that these cells were entering the cell cycle, the ones that we were looking at that went into mitosis so rapidly. And when we started to investigate where they were controlling this cell cycle, we found that they were actually, piezo was activating cells that were poised in G2 to push them into division and very rapidly bring back these cell numbers. And by investigating these different parts of the pathway, we found that actually cyclin B, what piezo is doing is activating cyclin B production. And cyclin B is a very important precursor that will activate CDK1 to go and cause cells to undergo mitosis. And the way that we found this out was by blocking these different steps in the cell cycle. And if we block CDK1 activity, we can just look at this intermediate buildup. And when you do, you can see that stretch activates cyclin B to be produced over time. This is cyclin B protein in the cell. Whereas piezo-1 inhibition will block that ramp up of cyclin B. So cyclin B could be produced either by blocking its degradation, activating transcription, or activating translation. And what we found by using transcription inhibitors is that what piezo is doing is really activating transcription. So if we block transcription, this ramp up in cyclin B does not happen anymore. So this finding was quite surprising to us because piezo is not known to be a transcription factor, whereas beta-catenin, YAP, and TAZ are. So this was a bit more surprising to us. And when you think about what piezo does, it's a stretch-activated channel. And this is the structure that was recently discovered of piezo-1. You could see it forms this propeller, which is a trimer. And it really sits in the plasma membrane and measures deflections in that plasma membrane to cause calcium entry. And that would cause these different types of events. So in this case, we wanted to test whether piezo was working in a canonical fashion by activating calcium. And so to do that, what we did was block calcium inside the cells and see if the cells could still undergo mitosis after stretch. And when we did, so we used BAP-AM, which chelates calcium inside the cells. And when we did, we found that this really blocks that stretch-induced mitosis as well as cyclin B production. So beyond that, we went looking for factors that could control calcium-mediated proliferation. And we identified a factor that was previously known to be activated by calcium, which is the MAP kinase MEK1 and 2. And when we block that, we block both the stretch-induced mitosis and the cyclin B production. So taken together, what we find is that stretch can really induce cells to undergo proliferation. Normally, cells grow and divide until they get to a density that they're really is optimal for their function. If they experience reduction in those numbers, either through apoptosis or if there's a wound or if they experience any stretch, this will cause piezo-1 to activate calcium-mediated influx, which will cause cyclin B to be synthesized through MAP kinase activation. This will then cause the cells to undergo mitosis so they reach these numbers that they prefer. Together, we've shown that epithelia prefer to be at a constant density. If there's too few cells suddenly from other cells dividing, they'll experience stretch, which will then activate these cells to go on to divide. Now, what I'll tell you about in the next segment is that when cells experience crowding, the opposing kind of mechanical tension, this will activate cells to undergo apoptosis or cell death through a process that we've identified called cell extrusion. And I'd like to end by thanking the people who've done this work in my lab and also my funding sources. Thank you.